Coming to our next chapter, cancer. So what is cancer? So cancer can be defined as disturbance in the growth characterized by excessive proliferation of the cells, uncontrolled proliferation of the cells, uncontrolled division of the cells, so that it has no relation with the physiological needs of an organism, right? So that is a definition of cancer. So what are the causes of cancer? Etiology of cancer is radiations, chemicals, viruses and free radicals. So these are the main agents which are responsible, main causes behind the cancer. So radiations like gamma rays, UV rays, X-rays. So exposure to these rays leads to cancer. So they are the physical agents. Coming to chemicals, chemical agents. So there are various type of chemicals like dimethylbenzanthracene, azo dyes. So all those chemicals, they can cause cancer, like aflatoxins. So these chemicals are capable of causing cancer. So these are called as chemical carcinogens. And the other reason is free radicals. So when free radicals are produced in our body in excess, those free radicals, they affect the DNA structure, and whenever DNA is affected, it leads to uncontrolled cell division, and that leads to cancer. And viruses, there are different types of viruses, DNA viruses and RNA viruses. So those are responsible for causing cancer. So these are the main reasons behind cancer. What are those reasons? Those are radiations, chemicals, free radicals and viruses, right? Now coming to different types of viruses. So we discussed that viruses are responsible for cancer. They can cause cancer. There are RNA viruses and there are some DNA viruses which are responsible for the cancers. Coming to RNA viruses, HTLV1 and 2 are responsible for the leukemias and lymphomas, and there is one more RNA virus, that is hepatitis C virus, which is responsible for the hepatocellular carcinoma. Like that, we have DNA viruses like Epstein-Barr virus, which is responsible for the nasopharyngeal cancer and also responsible for the Burkitt's lymphoma. So, and another DNA virus is hepatitis B virus, that causes the hepatocellular carcinoma, and another DNA virus is human herpes virus type 2, so that causes the cervical cancer, cervical carcinoma. So these are the types of viruses which can cause cancer, RNA viruses and DNA viruses, right? So whatever agent it may be which causes cancer, we call them as carcinogen. It may be a physical agent, or it may be chemical agent, or it may be a biological agent. So whatever it may be, we call them as carcinogens. Coming to molecular basis of cancer. So the reason for cancer is, so there are four mechanisms by which it leads to cancer. It may be because of gene amplification, it may be because of point mutation, it may be caused by promoter insertion, it may be because of enhancer insertion, so it may be because of translocation. So all these are responsible for the cause of cancer. So these are the molecular basis for the cancer. So what are those molecular basis? It may be because of point mutation, gene amplification, promoter insertion, enhancer insertion and translocation, right? So what happens in this? What are proto-oncogenes, oncogenes and tumor suppressor genes? We should know about these genes. So when you are discussing about the cancer, so proto-oncogenes, these are the normal genes which code for the growth regulating proteins. So that is the function of proto-oncogenes. So what are proto-oncogenes? These are the normal genes which code for the growth regulator proteins. And oncogenes, when proto-oncogenes are converted into oncogenes, that is, they are converted into cancer causing genes. So oncogenes are cancer causing genes. So these proto-oncogenes, they are transformed into oncogenes by various mechanisms, so that once it is converted into oncogene, cancer causing gene, it codes for the proteins so that are different from the proteins which are coded by the proto-oncogenes. So that's how it leads to, that's how it promotes the more cell division, more growth of the cells, tissue growth, and that leads to cancer. Coming to tumor suppressor genes. So genes which suppress the formation of tumors, so that's why they are called as tumor suppressor genes or anti-oncogenes. So oncogenes are cancer causing genes, and anti-oncogenes would suppress the cancer formation. Otherwise they are also called as tumor suppressor genes. Now here, whenever proto-oncogene is transformed into oncogene, and that leads to increase in the growth rate of the cell division, and that leads to a condition called as cancer. The same way, tumor suppressor genes which are involved in the would suppress the formation of tumor, would suppress the tumor formation. Now when they are transformed, when they lose their function, suppression capacity, then they are transformed into cells. So that is, they are inactivated. So when they are active, they can suppress the tumor formation, but when they are inactivated, they lose their function. They cannot suppress the formation of cancers. So these two, activation of proto-oncogenes and inactivation of tumor suppressor genes, leads to increased growth, and that results in the cancer. So that is the main mechanism behind the cancer: activation of proto-oncogenes and inactivation of the tumor suppressor genes. So what are tumor markers? So when a tumor is formed, those tumors, they release some factors. So those factors indicate the presence of a tumor in that particular person. So these substances which are released by the tumors, and they indicate the presence or growth of a particular tumor, is called as tumor markers, right? So they indicate the presence of a particular tumor. So they are acting as a markers for the presence of tumors. They indicate the presence of a particular tumor. So that's why we call them as markers, tumor markers. So the important examples for the tumor markers are alpha-fetoprotein and carcinoembryonic antigen and CA125 and prostate-specific antigen. So these tumor markers may be enzymes, they may be hormones, they may be proteins, they may be growth factors. So like that, they have different chemical bases, but when they are present on blood, they indicate the presence of a particular tumor. Right? When you take prostate-specific antigen, when it is present in excess amount in blood, more than its normal range, then it indicates the prostatic cancer. Right? So like that, CA125 indicates the colon cancer. Right? So alpha-fetoprotein, it can indicates the hepatocellular carcinoma. Right? So each protein, when it is present, it indicates the presence of a particular type of tumors in the body. So that's why you call them as tumor markers. So they are markers for a particular tumor. So this is about the cancer. So we discussed what is cancer and what is the molecular basis of cancer, what are proto-oncogenes, oncogenes, tumor suppressor genes, and why, what causes the cancer when there is defect in the proto-oncogenes and oncogenes.